Good morning, sir, and my fellow classmates. This is Ahana Majumder, and today I'm going to present before you the topic Environmental Biotechnology, along with my friend Goura Bibhu Ghosh. So, our topic of the day is Environmental Biotechnology. So, these are the contents that we are going to discuss today. Okay, so first we will talk about what is biotechnology. So, what do we usually think of when we hear the term biotechnology? Well, there will be multiple answers to this question. Biotechnology is the use of an organism or a component of an organism or other biological system to make a product or process. Many forms of modern biotechnology rely on DNA technology. Now we come to the topic environmental biotechnology. Well, environmental biotechnology usually refers to the use of microorganisms and other living systems to solve current environmental problems, such as the detoxification of pollutants and cleanup of oil tanker spills. Additionally, it is also referred to be used as in terms of agricultural environment as well as the use of biopesticides and the application of microorganisms to the mining. Now we come to this picture. Well, this picture gives a clear hint of the points that we are going to take note of when we talk about environmental biotechnology. Firstly, we have to take care of the manufacturing process, the waste management system, and finally, we will have to take care that the pollution is under control. All these are interconnected to each other. Everything should be taken under control and should be thought about with clear aspect about environment. Now we talk about role of microbial biotechnology in sustainable agricultural development. Well, sustainable agricultural development through microbial biotechnology describes how specific techniques can be used to generalize the metabolism of bacteria that optimize biologic improvement strategies and biotransport processes. The major strength of biotechnology is its multidisciplinary nature and broad range of scientific approaches. Recent advances in various biotechnological fields are facilitating the production of fine chemicals, recombinant proteins, biomaterials, and pharmaceuticals. So, here we can see a picture that gives us a clear hint of how these recycling processes are carried on. Firstly, the product is biodegraded, and then bio-based resources and waste is created, which is again processed through microbial biotechnology, and then the plastics that are bio-based and biodegradable are created, and again they are recycled and reused, and the same process keeps on continuing. Then we come to environmental biotechnology, the issues and implications that are there. So here we can see that these are the following points that actually focus on the issues: air pollution, ecotoxicity, water quality, and what not, ozone depletion, climate change, resource use, biodiversity loss, and water reuse. Environmental biotechnology is a multidisciplinary branch of science covering wide aspects of day-to-day life, where environmental pollution is one of the major challenges faced by environmental biotechnologists. Under normal circumstances, remediation of the pollution related to soil, water, air, all is naturally taken care of by recycling of the natural resources. However, recently, the widespread pollution beyond natural healing is a major concern. The increase in population and industrialization issues associated with agriculture, such as erosion of fertile soils and overuse of chemical pesticides, all are related to this. Okay, so here we can see that these are certain aspects related to environmental biotechnology. The things that we need to take care of when we actually create such manufacturing techniques for a better environment, such as antibiotic resistance, nature inspired biofuel production that will cause less pollution to the environment, electrochemical membrane filtration process, water reuse, biological wastewater treatment, and emerging contaminants. Okay, so now we come to the point bioremediation, or we can say environmental remediation by biotreatment. Bioremediation is used to clean up oil spills, or contaminated groundwater. Bioremediation may be done in situ at the site of the contamination or ex situ, which is away from the site of contamination. Bioremediation relies on stimulating the growth of certain microbes that utilize contaminants like oil solvents and pesticides for sources of food and energy. So plants, bacteria, fungi, yeast, all these contribute to bioremediation at a larger scale. Okay, so here are certain factors on which bioremediation basically depends. So when we talk about microorganisms, the microbial diversity, physiology, genetic competence, growth, and metabolism all are certain factors that we need to take care of. When we talk about environment, the temperature, pH, moisture, nutrients, and soil type are to be taken care of. And in terms of contaminants, bioavailability, recalcitrance, concentration, and solubility are to be taken care of. Now we talk about the role of microorganisms in bioremediation. Microorganisms can survive in all places on the biosphere because their metabolic activity is astonishing. Then comes into existence in all over range of environmental conditions. Now fungi, bacteria, yeast, algae that we can see here all contribute to role of microorganisms in bioremediation. So they actually lessens the inorganic pollutants, heavy metals contamination, antibiotic contamination, aquaculture waste, organic pollutants, petroleum oil contamination, aquaculture waste, and all such types of pollution causing terms. Okay, so now we come to wastewater biotreatment. Biological wastewater treatment harnesses the action of bacteria and other microorganisms to clean up the water. Now, wastewater typically contains a buffet of organic matter such as garbage, wastes, and partially digested foods. So, as we all know, that wastewater is a basic problem in today's world. So, these wastewater needs to be treated in a biologically processed way so that less pollution is caused and chemically active substances are avoided. Okay, so here are certain stuffs that we need to take care of. Firstly, biological treatment usually is divided into aerobic and anaerobic processes. When we talk about aerobic wastewater treatment, here comes the raw water that goes into the aeration tank, and in the presence of air, aerobic conditions can take place because oxygen is necessary for aerobic wastewater treatment. Then in the clarifier settler, the waste is settled and then treated water goes out. In this way, again, the wastewater sludge is being processed and recycling sludge carries on. Now here, there is another term, the MABR treatment, which stands for membrane alternate bioreactors. Now this is an alternate aerobic biological treatment process to MBRs, which is membrane bioreactors. And MABR is an example of a fixed film process, like a trickling filter, as opposed to a purely suspended growth process, that is one based on activated sludge, as is in the case for MBRs. So here we can see that the effluent nitrogen gets inside and then a baffle is created and biofilm is there where all this process carries on. Then the opening and the stopper is present at the top and below there are supporting net and membrane. From there, the waste and the N2 gas, which is the nitrogen gas, is carried out, and from above it is moved out as an effluent. And below we can see oxygen which goes out. The recycling pump is there which carries on this process while and while. Then we go for anaerobic treatment, which is in the absence of air. That is, a sludge banquet is present, the influent gets inside, and above the sludge banquet, it comes out as biogas and as effluent. Now, there is an important note that we need to take care of. Biological treatments often are implemented with additional treatment stages. Now, we all know that in household areas, chlorination is a very important thing. We often put chlorine to water in order to prevent any sort of problems related to water. Any pollution can be prevented, but that's only a primary stage. Above that, UV treatment takes place in scientific labs, And such filtration processes carries on along with carbon filtration, reverse osmosis, and ultrafiltration. All the researchers still continues to look for various other conventional methods for biological wastewater treatment. Okay, now we come to soil remediation. Many substances known to have toxic properties have been introduced into the environment through human activity. These substances range in degree of toxicity and danger to human health. Now, the certain activities also cause the removal of certain elements like hydrocarbons, halogenated organic solvents, halogenated organic compounds, non chlorinated pesticides and herbicides, nitrogen compounds, metals like lead, mercury, chromium, and radionuclides. All these are removed from the soil. Now, here the critical missile concentration CMC is taken into consideration. Dispersive action takes place, missiles are formed, and plants take up the important nutrients, and the rest of the contaminated part are being removed. So, here the brown part gives us the soil part, the gray part shows the contaminant, and water is shown in light blue color. So, here is this graph that is represented. Now we talk about solid waste biotreatment. Okay, so when we talk about solid waste biotreatment, from biofiltration to promising options in gaseous fluxes biotreatment, recent developments, new trends, advances, and opportunities provides an overview on the biological tools used for the treatment of the gaseous flux. Okay, so here we can see a pyramid is formed where the least favored are disposal, energy recovery, and recycling, and the most favored options are reuse, minimization, and prevention. Okay. Advantages of the biological treatment of solid waste include stabilization of the waste, reduced volume in the wastewater, destruction of pathogens that are present in the waste material, and production of biogas for our energy use. Okay. Now, biotreatment of gaseous streams. The biological treatment of air pollution depends on aerobic microorganisms, most mesophilic bacteria that feed on both organic and inorganic compounds in the waste gas. So here we can see the picture of biofilter, which is one of the main biological treatment technologies. So in biofilter, we will talk about the mechanism. In biofiltration, the polluted air is passed through a biologically active filter, or we can say biofilter. Microorganisms in the biofilter convert the air pollutants into harmless byproducts that are primarily carbon dioxide and water. Removal of contaminants is a multi step process. Now, the first step is the transportation of the contaminants into certain liquid phase, and then we will look into the stage 2, which is the transportation of liquid contaminant to bacterial cell in biofilm. And the stage 3 is transport of contaminant across cell membrane. Here we can see another picture that is bioscrubber, which is another important way to remove waste. Okay, a bioscrubber consists of a gas scrubber and a biological reactor. In the gas scrubber, to be removed components are absorbed from the gas stream by the wash water. In the biological reactor, the pollutants that have been absorbed by the wash water are biologically degraded. As we can see, the blue point gives us the hint of heterotrophic bacteria, and autotrophic are in orange dots. And here the odorous air inlet is present and the drainage system is also clearly available. Okay, now trickling filter scrubber. Now this is a very important thing because this is a blend of biofilter and a bioscrubber. Now, when we talk about trickling filter, it is very important to note that, as I have said, a biotrickling filter, it is a combination of biofilter and a bioscrubber. The bacteria responsible for decomposition are immobilized on a carrier or filter material. The filter material consists of a synthetic foam lava or a structured plastic packing. The surface must have a structure that allows biomass to bond to it effectively. The carrier material is constantly covered with water. This means that water must be uniformly sprayed over the packing. The polluted components absorb in the liquid film and are decomposed by the bacteria. As we can see, the rotating influent distributor is present and domed enclosure is also found here. Air inlet is there from where the air influent inserts there, and the air is also pressed upon from the opposite side. Thus, a trickling filter can be called a mixture of biofilter and a bioscrubber. That's all from my side. The rest of the slides will be explained by my friend Goura Bibhu Ghosh. Thank you.
Yes, the next topic is the biodegradation of hydrocarbon. What is biodegradation of hydrocarbon? In this, the hydrocarbons are the major constituents of crude oil and petroleum. As we all previously learned from the presentation, that the ability of microorganisms just like bacteria, fungi or algae to break down hydrocarbons is the basis for natural and enhanced bioremediation. In this, we can promote biodegradation. Amendments such as nitrogen and phosphorus fertilizer are often added to stimulate microbial growth and metabolism, just like oxygen, nitrate, or suppletes, and also some things that are added as electron acceptors to enhance biodegradation rates. The addition of hydrocarbon degrading microbial cultures to contaminated sites might also be considered, although the practice of bioaugmentation is common for hydrocarbons, owing to natural abundance of hydrocarbon degradation microbes. Aside from excavation, just like various forms of bioremediation from simple tilling of soils and land farming to biostimulation and common forms of remediation for hydrocarbon contamination. Compared to other technologies, just like bioremediation is very costly, is low costly, just like environmental friendly and often non-invasive. However, the time needed for full site cleanup can be longer and less predictable. So there are many methods that are being successful, so may not be guaranteed due to variability in site conditions. It is the most common approach for remediation and hydrocarbon of in contaminated environments. There are also some factors just like biosorption. Here we can see that biosorption can also be defined as the ability of biological materials to accumulate heavy metals from wastewater through metabolically mediated or physiochemical pathways of uptake. Here are some factors also in the in the given picture. You can see that the selection of biomass, then pre-treatment of biomass, then immobilization of biomass, then biosorption of granules, then granules dumped into metal contaminated solution, then desorption, then metal recovery. Now the next topic is the treatment of refractory organic pollutants in industrial water. Here are some compounds that are given here as like hydrocarbon, heterocycle, esters, organo, no, organosilicon, amine, phenols, halohydrocarbon, aldehydes, acids, alcohols, nitrates, and etc. In the given figure, you can see that there are catalysts. The next topic is environmental biotechnology in pollution detection and monitoring. As we see in recent years, environmental pollution detection and monitoring is being done by approaches involving biosystems. For this purpose, several group of plants, animals, and microorganisms are utilized. The Environmental Protection Agencies, just like EPAs, considered biomonitoring of pollution as a useful device to monitor environmental pollution from the point of diagnostic, preventive, and remedial measures. Now there are some criteria for biomonitoring of pollution. Number one is visual rating. Number two is genotoxicity rating, and number three is metabolic rating. Now the next is that what are the methods that are pollution in measurement and are briefly described as following order. Number one is general bioassays. Number second is cell biological assays. Number three is molecular biological assays, and number four is biosensors. What is bioassay? A bioassay is an analytical method that can be determine the concentration or potency of a substance by its effect on living animals or plants or on living cells or tissues. Here are some points that are given below. Number one is lichens in bioassays. Number two is mosses in bioassays. Number three, vascular macrophytes in bioassays. Number four is pollution induced peptides in bioassays. And number five is feces in bioassays. And the common important note is that protozoa and helminthes are also included as biasing among animals. Now, there are many more advantages also of environmental biotechnology. Number one is it helps to keep our environment safe and clean for the use of future generation. Number two is it helps the organisms and the engineers to find useful ways of getting adapted to the changes in environment and keep the environment clean and green. Number three is environmental biotechnology also help us to avoid the use of the hazardous pollutants and waste, and it can also affect the natural resources and environment. Number four is in environmental biology has an important role that to play the removal of pollutants. Number five is it is becoming an advantage for the scientist and also the environmentalist to find ways to convert the waste to US products. Now there are also many more disadvantages of environmental biotechnology. Number one is that what is blight in crops? In blight in crops, we can see that the implementation of biotechnology that has led to unfortunate development of blight in crops. What is blight? Blight is a type of condition that where the chlorosis or loss of chlorophyll green color of leaves. This may lead to withering or death of tubers and leaves since the plant would not be able to carry out adequate photosynthesis. Number two is lack of genetic diversity. In genetic diversity, although beneficial in terms of crop yield and production, offers to genetic diversity with populations. The genetic diversity is very crucial in long-term survival of plant and animal species. Larger the gene pool, the greater the chance that a particular species can survive due to its ability to cope with environmental changes and other pathogens. Number three is the loss of soil fertility. Bioenhanced crops can soak out of a lot of nutrients and vitamins from the soil. The high amount of activity that can threaten the fertility of the soil, it makes difficult for future crops to properly grown and harvested. The soil needs ample time to regain its fertility, otherwise it can have a detrimental effects on the production of food. The excessive drain of nutrients from the soil leads to farmers obtaining fertilizers which may be very detrimental in the long run. Number four is that the gene manipulation techniques can be misused. Yes, the knowledge that can be produced, enhance animals, humans, and plants has led to humans using such methods more frequently out of greed. The use of biotechnology to make children better with the parents' preferred phenotype has become common, even if they are not at risk of developing any underlying diseases. These have led to the banning of designer pebbles in countries such as United Kingdom. Number four is biotechnologies are also very costly. Number five is reduction in human genetic biodiversity. And number six is used for destruction where, for example, that is coronavirus. Now the conclusion. Considering these pros and cons of environmental biotechnology, it is imperative to say that biotechnology is a very interesting field which could have for reaching advantages. However, considering that even make a single mistake would be harmful to a number of generations ahead, scientists need to be extremely careful with applying biotechnology. And it is also obvious still it is in its infancy and can be used to make changes that may lead to the next big revolution. The pros and cons of every application of biotechnology should be weighed, not only 10 but 100 times before a biotech product of application reach the markets. Now here are some references that I have taken help from to make presentation. Go www.google.com, Biology Class 10, 11 IC book by V.V. Rastogi, and Class 12 book also by V.V. Rastogi. Thank you.